Thank you very much. Hopefully you can see that first page slide. I'm not hearing anything saying not, so there we go. So yeah, thank you very much. Really talking about some of the work that we've done over the last 4 years. I wanted to start by acknowledging everyone who'd been involved in it, because obviously, you know, I'm just the mouthpiece for it, but there was a large team around it. I won't name check everyone, but they're all pictured here. And yeah, it's really just thanks to everyone who's been involved in it. Nello gave a really nice overview of the sort of reason why we want to control parasites. When I was thinking about it, you know, when we think about the sort of things that livestock carry, there's a whole range of these things that cause you know, health and welfare issues and production losses. We decided and wanted to focus on the roundworms, so that is where the focus of this is. And really, as Jill said there, we really were looking to see what the impact or how these featured in transported or animals that were sold and in wildlife. Why does it matter that we have a bunch of species? We know that anthelmintics are licensed against all of the different ones. We know there's about 11, 12 different species of roundworms. I put this up as much just because I had all of the images and I thought it made a nice backdrop for it all. But it also highlights the fact that they come in a whole range of shapes and sizes. Their susceptibility to anthelmintics differ, but also so does their ability to develop anthelmintic resistance. And I think that'll become more and more relevant as the talk goes through, hopefully. Yeah, treatment options may differ. So it may be that there's opportunities for using narrow spectrum anthelmintics for the sort of blood feeding parasites. Their ability to overwinter on grounding in the host differs. Their climatic preferences differ. So there's lots of factors that can influence what happens on farm and what happens in the host as well. The two tables at the bottom really just highlight again why we care about species, because you know, we have as I said, a whole range of different species there. They have different relative pathogenicities, and parasites in sheep like Homunculus and Telodorsagia are very pathogenic. The problem is they're also the ones that are most likely to develop resistance, and the ones that we see resistance develop to in the UK and globally. In cattle, it's slightly different. The pathogenic species are not necessarily the ones that are currently selecting for resistance. But again, there's none of these things occur in isolation, and there are interactions between these. So when we're thinking about movements and animal movements in particular, you know, the last sort of data we could find was that there's something like 1.5 million cattle and 10 million sheep sold at markets each year. Of course, not all of those will move from farm to farm, but you know, a proportion of those will. NSA and Scops did a survey around the same sort of time as that, and were asking farmers whether or not they felt adequately informed about their worming history, and half of them did. But when we have done, and I know Eric has done the same, when we've done questionnaire surveys, we know that, and this is relatively consistent over the years, about half of farmers don't quarantine, treat their animals, and half of those probably administer a treatment that is likely to be suboptimal. So, you know, there is an issue of how parasites can move from farm to farm. And additionally from that, there is also increasing interactions with wildlife, with livestock as well. And again, because of habitat loss and increasing areas being used for livestock, you know, again, we have these interactions. So the aims of the project were really, we hadn't quantified or qualified what was in animals that were moved or really within the wildlife either. So what we'd set out was to try and get a better handle on what was being transferred in sheep and cattle, and also within deer, in respect to what roundworms they were carrying, as well as the sort of white drench resistance profile. Unfortunately, we don't have markers for more than the white drenches, so that we use that as a proxy. And also the other thing that we wanted to do is get messaging across. So we were involved in developing some tools to get messaging away through to stakeholders. So I'll talk a little bit about that. Okay, so the workflow as it stood, yeah, we went about, we gained access to marts and got contact details. We went out and we went and collected fecal samples from pens. It was not done in a random fashion. It was done very much on a opportunistic fashion of what was there. We went into pens as animals were being moved into the ring and then had to be out again. Obviously, with sheep, that's a little easier than it is with cattle, but thankfully the marts staff helped us with these, making sure that we stayed safe. We were able to do egg counts on those samples. For those that we had sufficient parasite material from, we were able to extract the DNA, and that allowed us to look at twofold. We were able to look at the ITS2 region of the gene, and basically that just allows us to look at what species were in there. And as I've said, we were also then able to look at the beta tubulin, and that again is the sort of place that the white drenches act upon, and mutations within this region allow for resistance to develop. And we were able then to sort of get an idea of both prevalence, but also where the white drench resistance resided within it. And as I say, this gave us a snapshot of the sort of prevalence over time. So if we start with the sheep, the egg counts, we were able to look at about 441 samples from about 13 different marts. And we did egg counts as the image on the top right hand side shows. For those of you who've seen them, you know you can pick apart the sort of strong eye eggs apart from things like Nematodyrus and Nematodyrus batus. So we did this, and yeah, 90% of those samples were positive for egg counts, and the sort of average across the board was around 224 eggs per gram, but that actually ranged from no eggs being identified in those 10% all the way up to 11,000 eggs per gram. To put that into context, that animal that was putting out 11,000, or a group of animals that was putting out an average of 11,000 eggs per gram, you know, conservatively, that could be then putting out 11 million eggs per day onto pasture. You know, so that's a huge contamination risk for wherever that is if they're not appropriately treated with a quarantine treatment. Again, when we looked at Nematodyrus, there was slightly less, only about 2 and 3 had positive counts. Their egg counts were quite low, and the range was low also. That probably is more of an artifact of the time we collected them, because we collected them from summer through to winter. Unfortunately, COVID got in the way. We had plans to go in spring, but obviously many of you will remember that yeah, spring of 2020 was not a time for going out collecting samples. So unfortunately, we never got back to being able to do it. When we look at these in a more structured way, one of the questions that always comes out is, well, okay, so you've got a bunch of zeros in there, and you've got a bunch of low counts, and you've got an average of 224. You know, why are we treating these? You know, and one way of looking at this is actually saying, yeah, we have a large portion that are low in there, but we also, we have a quarter of those farms where it's medium to high. As stated before, we have high levels, it's really high levels in some of those. And not only it's not just on counts, but it's also on what you're bringing in and on to those farms, which is important. And I raise this just to highlight that, the average is 224, which is about the point at which the sort of guidance would suggest we treat animals. If we look at what the sort of roundworm species of those samples looked like, I've put this up really just to show you that each of those bars is a single sample. Obviously, you can't really see very much in that, but it just highlights that we found a good range of different species. We found a large amount of them, but I will transfer it to this, and this is the average across all of the samples. So as you can see, the largest proportion there was Tila dorsagia, the brown stomach worm, and that, as I said before, is the species that actually is the most likely to carry resistance, and I'll talk a bit more about that in a second. We had about a quarter of the samples that were Trichostrongylus, again, a pathogenic species, or a group of species, and a moderate level amount of large bowel worms, and this agrees with what we would have seen in previous studies where we've looked at the sort of epidemiology of these things. When we looked at the white drench resistance, these charts here highlight the different mutations that we see that are associated with white drench resistance. So we can have single mutation changes at 3 different areas. The commonest is generally considered to be P200, but we see them at 198 and 1672. And this is what we found when we looked at the species. As you can see on the far left of that's of the slide, we had red for resistance and green for susceptible, and you can see that bottom left-hand chart is really exceedingly red, and there's not a huge amount of green in there, and that is probably what we would have expected. Interestingly, though, we also found mutations in the other two areas too, and again at quite high levels. So as maybe we would have expected, white drench resistance in T. circi is quite far advanced, and at some point there's no susceptible genes in those populations at all. When we look at the other two species, so we look at Homunculus, the barber spole and Trichostrongylus, again we see that resistance is slightly less developed, but interestingly we see that the genes are slightly different, certainly in Homunculus, and it favors the sort of 167 mutation. And again, with the Trichostrongylus, we see almost none of that, but we see lots of 200. Why does that matter? Because actually there is some work that suggests that where those mutations lie can impact on the sort of phenotype that we see. So some of them select for a stronger gene resistance genotype than others. Obviously, once you get the whole population containing genes for resistance, then the product is not going to work. So yeah, we found varying levels in the different species. The T. circ was at one end, and then T. colubriformis and Homunculus and T. vitrinus were at the other end of that spectrum. So what we see is that the drugs very much can have different effects, even with white drenches across the species. So really, just for the sheep, what the take home message is there's a modest average count across the piece, with some carrying huge burdens and others carrying less. But again, it is important that all of them get treated on there. Co-infections are common. So as was seen from those charts, which is good. The worry is that we get monospecific or monocultures of particular species, which it doesn't look like we are. So that is good. We have variable white drench resistance prevalences, very high in T-circ and low in the others. The different mutation combinations in the different species, again, yeah, is that an artifact of the selection pressures that they've gone on? Because, of course, historically, different species occur at different times, which means that they will be exposed to drugs at different time also. But it does reinforce that sort of quarantine recommendation that we need to ensure that effective quarantining and testing are carried out in sheep that are being moved from place to place. Okay, if we're looking at cattle, what we see, again, we gathered slightly fewer populations here. We only got just over or just under 350. 3/4 of them carried a positive counts. And the range, again, went from 0 up to 750, so not as high as in the sheep, but a lot of people would argue that they, the top end of that is still quite high. The averaged at 24. Again, that is when you would look at it as not being very high, but actually in comparison to the sort of average that we'd expect in cattle, that's a decent figure considering the amount of feces that these animals put out. Again, the chart just highlights the different marts and what the sort of spread of the accounts actually is. Interestingly, we didn't find very much nematodirus within those samples, but again, that may well be an artifact of time. When we looked at a species composition, again, we were able, because of the smaller number that we were able to get sufficient material out of to look at species, we can bar chart it. The predominant species are Cuperia, Tiaxii, and Ostotargia. And again, if we look at that can be seen in the pie chart as those as being the predominant species. So, the most prevalent was Ostotargia. It was found in almost every sample. The overall prevalence was about 44%, as can be seen in the pie chart, but that ranged from 0 to 98. So that it was found in almost every sample. Cuperia was again found on average about the same sort of amount, but it was only found in about 3/4 of the samples, and Tiaxii was found in about half of the samples. So yeah, these species are important. And again, understanding what the species composition it is made up of is important, because we see much more resistance in Cuperia with the MLs, with the macrocyclic lactones, than we do in the other species. But we are starting to see resistance in Ostotargia. We've had white drench resistance in it where there's been total clinical failure. So we need to bear this in mind when we're looking at it. When we looked at the white drench mutations, again, we did find high levels of white drench resistance, but when we did find it, it was at saturation within in some of those populations. So again, we can look at P200, P198, and 167. Interestingly, there was another mutation that was observed, and it appears that it is correct at position 199, but we need to do more work on this to look at it. But as you can see, within the Tiaxii populations, we had mutations within the same population at two points. So again, there are populations out there where the white drenches won't work. And again, off the back that we have quite high levels of ML resistance, that is something for us to think about, and it is a time where we can do something about it. Yeah, again, as with the sheep, the average count was lower than in sheep, but still was not 0. Co-infections are common. We do see white drench prevalence in cattle parasites. but we are seeing it in multiple species, which is a bigger concern. We do now have that sort of baseline to monitor this going forward, and it is something that we do need to bear in mind. The different mutation combinations present, as I say, is important because of the sort of phenotype that might be there. I think looking at that, you know, the sort of risk is that it's at an early stage, but we are at a point where, you know, appropriate advice and intervention could help this slow this resistance development down. So again, we need to think about how we get messaging across to cattle producers. Excuse me. Okay. In deer, we were able to look at both wild deer and farm deer. We followed a very similar process workflow. We were able to get samples from, as I say, from farmed, as well as deer that were culled in it for a study for looking at E. coli. So we were able to get access to some of those samples. So what did we find? If we look at the wild deer populations first, again, we were able to look at a large number of these. We're looking at almost 650. 80% of them had a count, and again, they had a modest average egg count at around 33. But you know, the high point was, you know, almost 2,400. So again, large numbers in some of these animals. We were able to look at red roe, Sika, and Fala. So it looked like it from the samples that we got higher counts in red and roe in comparison to the Sika and fallow. But again, we had more red and roe in our data set. Again, we were able to look at the species composition, and yep, that top chart is just, yeah, for making probably making you feel a little nauseous, the magic eyes, but the sort of chart at the bottom highlights that within samples, you know, we had lots and lots of Oesophagostomy, lots of Osteotarchia leptospicularis, but also we found species that we would associate with cattle and sheep in there too. So we found Osteotarchia osteotargi, as well as some of the more sheep-related ones, so Tila dorsagia and the trike species. So again, they are carrying wildlife. They are carrying livestock species within them. So again, something to bear in mind. If we looked at the farm deer, we got a very similar output, higher count, on a higher prevalence of positive counts, but again, the mean egg count was about the same, and the average was slightly lower. Sorry, the maximum was slightly lower, but again, you know, we saw that these things carried large numbers. Excuse me. What was interesting is that when we actually looked at the species composition of these, obviously these are they're farmed red deer, so they don't have the opportunity to go roaming around as their wild compatriots do. But again, we saw large amounts of Oesophagostomy, Osteotarchia leptospicularis, and some Succuperia there. So again, it was interesting to see that they do carry these livestock species. And interestingly, when we looked at the profiles, we didn't find very much in the way of white drench resistance, but we did find it in both the wild populations and the farm populations at the sort of position 200 and 198. So again, they are carrying resistance around with them, albeit at quite a low level. So yeah, in summary, for the deer, they are infected with a wide, certainly the wild deer are infected with a wide range of livestock parasites. The fecal egg counts are modest, but as I say, some of those individuals are much higher shedders. The wild red and roe deer appear to be the most commonly infected, and white drench resistance was identified in both. And again, the question we wanted to know is, actually, the original question we posed for the deer was when we were looking at white drench resistance in Nematodyrus, and we pondered whether we when it was found in areas, whether or not deer were one of the factors that were spreading a resistance around. It appears that wasn't the case, because we didn't find very much nematovirus in the wild deer. But again, it is something to bear in mind that these may or may not spread resistance around. The counter argument to that is that they carry a lot of species, so they may well act as refugia if they've got large amounts of susceptible parasites in there too. Okay, I know I'm getting almost to the end of it all, so just to briefly talk about some of the knowledge exchange stuff that we've been doing. We were able to look at updating a model of the cost of resistance in the UK, and we were able to assist in the development of some of the materials for stakeholders. So we were looking at developing a decision support tool for multiple anthelmintic resistance. We involved in a sort of fact check tool and developing materials for SCOPs and health and welfare pathway. And I'll show a little bit of that. So the model output, we were building on a publication that was put out in 2020 by Johannes Charlier and many others from across the UK. From across Europe, and we were able to use this model to look more closely at what the situation is in the UK. The table at the bottom really just highlights what the sort of cost of resistance is, and again, it's building up an idea of what resistance costs the sort of UK livestock industry. Again, you'd be able to have a look at these, but within cattle, in essence, it is the MLs, where we see huge amounts of the losses in there. So it's about 1.7 million in loss to the dairy cattle, whereas in sheep we sort of see benzimidazoles as the highest point. The number in parenthesis in the brackets behind the sheep one at white drench resistance is losses to nematovirus batters. So there's about 190,000 per year just lost in just to white drench resistance in nematovirus batters alone. So as you can see, that the costs are not insignificant at about 5 million per year. Yep. Again, it was really just to highlight that some of the outputs from it were that we were able to sort of look at different resources. So there's an app, the Fact Check app there, which is really a resource for people who are doing fecal egg count reduction tests or monitoring egg counts, to put it in and to give them a nice visual view on it. So again, if people are doing it, it gives advice on how to collect samples, and it's gathered together some of the materials from this and some of the materials that were put together for the health and welfare pathway. So again, the video at the bottom is on the SCOP site, and the link at the bottom really just highlights where you can find these materials. So if you or clients are your clients are doing fecal egg count reduction tests or drench checks, then actually there are materials on how to collect samples and what it should look like. So again, I would ask you to go and have a look at these materials, because they are there for everybody's use. So in summary, yeah, we had a comprehensive and up-to-date sort of data on the prevalence and scale of roundworm infections in sheep across the UK, but in this case in traded sheep and cattle, as well as in wild and farm deer. We had again updated on the white drench prevalence in UK stock, and again that highlighted that white drench resistance is exceedingly common in our pathogenic roundworm species, particularly in sheep. And this is off the back of knowing that we have lots of multiple resistance in this, but we do have material there so that we can look at other classes of resistance if and when markers become available. In Ostatagia, yeah, it appears that there's currently less white drench resistance, but where we do see it, the sort of prevalence is high within those populations. We see the early stages of white drench resistance in Cuperia, but we know that ML resistance is a problem within this species and within this host within cattle. So it may be that white drenches get used more readily for cattle, and the selection pressure for pushing up resistance has the capability to make this a bigger issue. And this is certainly what has been seen in places like New Zealand and Ireland, where we see larger prevalence of multi-species resistance to multiple classes. So again, I think we're at a point where we can do something about it, and now is the time to act. I think ongoing knowledge exchange is essential. Yeah, and we need to get this in various different formats, so to encourage people to look at it and act upon these things. So that just really leaves me to say thank you for your attention and to hand back to Jill.